All right, so good day, everyone. Let's move on to microbial growth. So these are the learning objectives for this lesson. And let's talk about first about binary fission. So microbial growth refers to the increase in the number of cells in the number of say cells or the increase in the number of cells, not the size of individual cells. So the most common way bacteria reproduce is through binary fission. It is a simple but efficient method that allows rapid population growth. So in binary fission, a single bacterial cell duplicates its DNA and then divides into two identical daughter cells. So key points in here, reproduction is always asexual in prokaryotes. So genetic variation still occurs due to horizontal gene transfer. Example are the conjugation, transformation, and transduction. Why is it important? This rapid growth allows bacteria to quickly adapt and colonize new environment. So, steps of binary fission. We are now breaking down binary fission into three main steps. Number one, growth. So the cell increases in size and prepares for division. Number two, DNA replication. The bacterial chromosome is copied at a specific point called the origin of replication, and duplicated DNA moves apart. And number three is the cytokinesis. The cell divides into two daughter cells guided by a protein called FTSZ or Z, which forms a Z or Z ring to pinch the cell in two. So this process ensures that each daughter cell receives an identical copy of the genome. So in here, you are looking at the visual representation of binary fission. So the parent cell grows and copies its DNA. The DNA moves to opposite sides. The cell divides into two identical cells. And this simple yet effective process allows bacteria to multiply quickly. Now, microbial growth in generation time. Generation time or doubling time is the time it takes for a bacterial population to double. So fast growers like E. coli, Escherichia coli, can double every 20 minutes under optimal lab conditions. Slow growers, some bacteria take days to double in extreme environments. So why does this matter? Knowing the generation time helps scientists predict bacterial population growth, which is crucial in medicine, food safety, and biotechnology. Let's go to understanding generation time. The growth rate of bacteria varies. So, Escherichia coli or E. coli can double every 20 minutes in a rich nutrient environment. Some bacteria in harsh conditions take much longer to divide. So, implications: fast growing bacteria can cause rapid infections, while slow growing ones may persist in difficult environment. Next is calculating generation time formula. The formula for calculating population size during bacterial growth is um, n or nn is equal to NO2 raised to n, as you can see on the board, wherein n um, n here number of cells at any generation, and O is initial number of cells, and n smaller n number of generation. So this formula helps us predict bacterial growth over time. Let's have a sample problem in here. We're going to apply this formula with these two or three samples. But let's see. Problem one. If the generation time is 30 minutes, how many bacteria will be present after 24 hours if we start with cells? Okay, you just need to substitute. And the answer for number one is 2.81 raised times 10 raised to 14 cells. How about number two? If the generation time is 30 minutes and we start with 300 bacterial cells, how many will be present for 12 hours? Okay, the answer is 5.03 times 10 raised to 8 cells. Bacterial populations grow exponentially, which is why infections can spread so fast. So, in conclusion, binary fission allows bacteria to rapidly reproduce. Generation time determines how fast bacteria grow, and understanding microbial growth helps in disease control, food preservation, and biotechnology application. Okay, that's all for today. Thank you very much.